Hello, thank you everyone for joining us in our first SE3D educational webinar. We're hoping to start these as a series, so thank you for joining. I'm Sarah. I'm one of the engineers here at SE3D, and Oliver, who's also in the call, will be doing a demo for you guys later. So I'll go ahead and share my screen. Just a desktop. So today we're going to be talking about bioprinting. So I'll spend a bit of time giving background on the field. Then I'll talk a bit about what we do here at SE3D. And then Oliver will do a demo print for you guys. So a bit of background. Many of you are probably familiar with 3D printing. So 3D printing techniques were first developed in the 80s as part of a wave of additive manufacturing. Today, they are largely used to support the fast prototyping that we have come to know as part of the design thinking methodology, and are by and large printing plastic filament. On the other hand, bioprinting is printing biomaterials. So usually that's a soft gel or liquidy material, as most biomaterials are, and the ability to print these biomaterials allows exploration into cell and tissue engineering. So as opposed to making quick prototypes or models, bioprinting allows you to print real living structures that you can then perform tests on in vitro and hopefully eventually even transplant into humans and animals. So how does a bioprinter work? In terms of mechanically, it works quite similar to a normal 3D printer in that you have movement in the X, Y, and Z directions so that you're able to print a 3D structure. In terms of what you're printing, you're not printing a plastic filament as you would be with a normal 3D printer. You're printing a bio ink. So what that looks like in the case of maybe printing tissues, it would be printing a bio ink or hydrogel that supports the cells as you're printing them, and after they're printed, that can then be printed in layers to create tissues in different structures that you've modeled. So this photo here is from an image from Organovo. They, in partnership with Invitech, created a printer called the NovoGen that uses the process you can see in this image. So they print these biospheroids in layers, and each of the biospheroids is an aggregate of thousands of cells. So they're printed on layers of biopapers that are made of the same kind of hydrogel I talked about before that supports the cell functioning. And so they're printed in layers, and the biopapers either dissolve or are removed, after which the cells naturally rearrange themselves and do the work for you. So applications for bioprinting are plentiful. The holy grail of bioprinting is obviously the capability to print organs. So companies like Organovo are working on reaching this milestone, and they were not too long ago able to successfully transplant liver tissue into mice to cure liver failure. So maybe soon they'll have a functioning liver, we'll see. Biofuels is another promising application. Researchers were able to demonstrate that microalgae could be capsulated in 3D alginate scaffolds. So this obviously looks promising for using bioprinting to create biofuels in the future. Drug discovery is another large application. The mere ability to print 3D structures rather than testing on traditional 2D cell cultures in itself creates a much more realistic application for testing drugs. Meat production is another application. One of the co-founders of Organovo went on to found a company called Modern Meadow that has been working on creating synthetic leather and meats. So they spent $100,000 to create and successfully bioprinted a cut of meat. So that's quite expensive meat for popular consumption now, but in the future that could, meat production and just general food printing is definitely promising exploration. And organ-on-a-chip devices is also another application of bioprinting. So organ-on-a-chip devices are 3D microfluidic cell culture chips that stimulate the mechanics and physiological responses of organs. And the benefit of them is that, one, that it's 3D, so you have the advantage of simulating something in 3D rather than a 2D cell culture, and you also are able to reduce the size of what you're testing significantly. And so 3D printing and bioprinting have a lot of, I think, promise in contributing to organ-on-a-chip devices. Harvard researchers were recently able to, in 2016, 3D print the first organ-on-a-chip, or the first 3D printed organ-on-a-chip device. That shows a lot of promise for taking away the barriers of producing these devices. So here at SE3D, we're committed to bringing bioprinting to the classroom, so in education and academia. So we have some examples here on screen of what that could look like. One on the left was at a school in Pennsylvania that the students were working on printing bioactive glass with our bioprinters, and this was part of an independent research project. And on the right, we have a school that's local to us in California that our bioprinter was used as part of a whole biotech class. And so in this image, the students are printing enzyme assays just as part of the curriculum for the course. And so when the founders of SE3D, Maya was here, created the printer, they wanted to design it for the classroom. So some of the features that come with that are that it's compact and portable, has a multi-user build platform. You can print both gels and liquids, and you can do liquid handling, which allows multiplexing. And yeah. So along with our printer, we offer curriculum to coincide with it for the high school level. There are a number of tracks in our curriculum that are biotechnology, green technology, tissue engineering, and we're working on our food science track. And along with these tracks, we have different lab modules. So one covers just the basics of the printer and 3D modeling to get you familiar with that. And then other lab modules cover proteins, enzymes, bacteria, and algae that teachers have been using just to complement what they're already teaching in their classes. Hello. So today we're going to be printing a scaffold out of a material called pluronic that I'll talk about in a bit. So I figured I would give some background on tissue engineering. The general process is that cells would be taken from a biopsy from an individual grown in vitro and then potentially grown on a 3D scaffold, like potentially like the one that we're printing today, to generate a graft that you could then put back on the patient. So a little more on pluronic. It is a hydrogel that's non-toxic, it's biocompatible and biodegradable. It's thermosensitive, so when it's chilled, it's liquid, and as it reaches room temperature, it's a bit more jelly, and that's the state it'll be at when we're printing today. So we already have prepared everything to just run the print to show you guys, but generally the process for doing this would be that you would make the 3D model. You don't need to be a professional engineer to do that. There are plenty of resources like Tinkercad available to make that easy. You would then load the materials. So we use disposable syringes. So you would just fill your syringe with Pluronic, load it into the printer, and then load the file and print. So another module and lab that we offer involves enzymes and encapsulating the enzyme reactions in gels. So the materials like alginate, agarose, and collagen are promising for being able to encapsulate reactions. And Oliver will also be showing you guys that today. So in terms of assays and printing biological arrays that you could potentially, again, use like agarose or alginate to encapsulate, the process, again, would just be setting up the printer and printing the gel and then the bioentity. And once you've encapsulated the bioentity in whatever gel you're using, like potentially alginate, you can then print substrates to catalyze your reaction and view it in potentially like a colorimetric assay, like the enzyme assay that we have prepared. So as I said, this is our first webinar. We'll hopefully be offering many more. In terms of our, we host a number of workshops as well that cover a number of different labs. So we have an enzyme lab, algae, E. coli, chocolate printing, and just general 3D modeling. We also offer, so we both, as I said before, produce bioprinters, and we offer bio kits that complement the curriculum that we provide. So there are the bio kits have the materials for the enzyme or Pluronic and the different labs. So everything from the photosynthesis to starch degradation and everything you would need. And if you have any questions, you can contact us at our email or go to our website. And if you have any questions for us or for me or for Oliver as he does the demo, you can also put that in the chat in Zoom. So I'm gonna hand this off to Oliver right now.
All right, so I'm going to be doing Pluronic print. So I've already loaded it in the syringe here, as you can see. So I'm just going to go ahead and load it into the extruder unit of the bioprinter. And the first step is to prime the syringe for printing, just to make sure that the material is flowing out. So I'm just going to manually extrude until I can see Pluronic exit from the needle tip. Next, I'm going to set the Z0 position in order to properly calibrate the bioprinter. To do that, I'm just going to move the extruder unit down until it touches the bed. And a good way to measure this is just to see that you can slide a piece of paper under the needle tip. So now I'm ready to print, so I'll just load my Petri dish here. Glenn, to answer your question, yes, we do have limit switches, but so they're used to set the position when you home the printer. But if you manually set the command to say move the extrude unit, 1 meter, it won't have a switch to detect that. So it's up to you to make sure you don't send an improper command. But yeah, so the scaffold is done printing now, so you can see that it kind of has a 3D structure. So you can see that it has multiple layers and there's an infill in the middle. And I'll also show an example of an array print. So like Sarah was saying, you can use this print to print liquids such as calcium chloride and alginate to create encapsulated arrays. So here I just have a syringe filled with dyed water just to help visualize the process. So I'll go ahead and load that. And then once again, I'll prime the syringe by extruding until I can see droplets begin to form. And then once again, I'll set my Z0 by making sure I can hold the piece of paper under the needle tip.
To answer Glenn's question, the bed material is glass.
And I'll also secure the syringe in place with these clips. And then place my petri dish and then start the print. So here you can see that the printer has created discrete droplets in a 3x3 array.
No question. Maybe address
So you can see how this one is what we call a continuous print, in that it's continually extruding in lines to create a 3D structure. Whereas this one, we've created discrete droplets, so instead of extruding continuously, it almost serves as a liquid handler. Let me screen share the software that we use to control the printer so you can get an idea of what it actually looks like. So you can see here how the print is loaded, so you can see sort of a preview of what is actually going to be printed. So, wait, can you see?
Can you see the software?
How do we change the window?
This one?
Can you see this one?
No. Desktop. Just share the desktop. Oh, that one. There you go.
No. So in this one,
very tricky. The other one.
So in this screen, you can actually get a move by move of what is actually going on. So you can see how it moves from the corner over to the starting position of the print and each subsequent layer. And then finally the extruder moving to the back once the print is completed.
And so just to, before we close out, I wanted to see if any of our participants have any other specific questions. And if not, then thank you guys for joining us in our first webinar. And we hope you guys will join us again in the next one. And if you do have any questions that come up after this, we will, you can email us or contact us over our website, and we'll also provide the link for this video. A question from Glenn. What is the purpose of encapsulating live cells in a soft polymer? And the purpose would be that you can visualize the reaction and to answer this.
So let me just quickly answer that question. So I think one of the main goals of encapsulating any living organisms is, one is you could isolate the living organism in that space, right? So for medical applications, that could be potentially utilized for drug delivery applications, or maybe cell delivery into specific locations. But for the purposes of testing, it makes it easier sometimes to isolate different species independently. So in a lot of the testing and assay platform, sometimes encapsulating the cells would help in that sense. So for us, it just makes it a little bit more convenient to work with different cell types and different compounds when you isolate the cells within a polymer and the gel. So that's one. Other applications, obviously, if let's say it's a cell that actually grows in a three-dimensional space, like a tumor cell, then obviously it would make sense to grow the cells in a cephalroid rather than growing them in a 2D kind of a culture plate system. So that would be most of the applications for encapsulating cells within a specific gel material. Great. Thanks, Glenn, for joining us. As Sarah said, if you have any additional questions, feel free to reach out, and we'll definitely share the video with you as well.
Thanks, everyone.
Thank you. Bye.